I'm going to talk about fluidic force microscopy and how you can use it for CRISPR and also some omics. So there is definitely some connection there. And the first point that I would like to quickly introduce is what is FluidFM even. So it stands for fluidic force microscopy. And this is basically a method that is combining microfluidics and some principles of atomic force microscopy. That's how it got the name, but actually it cannot do microscopy. So it's more of a fantasy name. And what it can do, however, is to interact with cells in a force-controlled manner, which means that we have these tiny cantilevers that you can also see on the picture. These cantilevers are roughly 200 micrometer long, and they contain a microfluidic channel. Now, these cantilevers can interact with cells, and this interaction is something that is constantly measured through the force control feedback loop. And at the same time, in the microfluidic channel, we have a liquid that is dependent on the experiment itself. And this liquid can either come out of the cantilever by the application of positive pressure, or it can go into the cantilever by the application of a negative pressure. And these different pressure ranges then allow for many different applications, also depending on the geometry of the cantilever. Now, this technology was developed here in Zurich in the ETH Federal University, and it's patented. So only Cytosurge is developing it in the whole world. And let's look into the geometry a little bit. So I mentioned that the cell interactions depend on the geometry of these cantilevers. And indeed, at the tip of the cantilever, we can have different structures that allow for different kind of applications. So in one way, what you can see on the left side is what we would call the micropipette. In this case, we have a flat cantilever, and at the end, a very simple circular aperture. Now, this circular aperture can have different sizes. I can see it's in the range of 2 to 8 micrometers. And what we can use this for is that we can manipulate cells. And by manipulate, I mean we can pick them up and place them somewhere else. By picking up, it works with a negative pressure. So we can just simply approach a cell, apply a negative pressure in this channel, and that simply mechanically fixes the cell membrane to this cantilever. The other very important application is happening with a nanosyringe. In that case, as you can see, the geometry changes from a flat cantilever to a pyramidal cantilever. There at the end, we have a pyramid with a sharp tip, and next to the tip is the tiny aperture. This aperture is smaller, around 600 nanometers. And the reason for this kind of geometry is that with this kind of cantilever, we can go into cells, which means that we can actually pierce the membrane. That's why the sharp tip is still there. And once the cantilever is inserted into the cell, then through this tiny aperture, we can either inject material with a positive pressure or take material out with a negative pressure. There is also a nanopipette, which is a combination of the two. This is mostly used for for surface patterning, which I'm not going to talk about that now. But the point is that these different geometries make it possible to have different applications, as I mentioned. So with the micropipette, we can pick cells and place them somewhere else. Now, this picking also means single-cell force spectroscopy. Since, as I mentioned, we always have a readout of the force between the cantilever and the cell, when we take a cell from the surface, we can constantly measure the force that was applied between the cell and the surface. And this way we can measure adhesion forces, for example. And on the other hand, when we go into the cell with the nanosyringe, then we have the possibility to make material enter to the cell or take material out of the cell. Now, how does it look like in the lab? So all these manipulations are made possible by what we call a FluidFM Omnium, which is it's a device, basically, as you can see also on the picture and on the little video. So here, what we have, these little probes, as we call them, these are the parts on which the cantilever is mounted on the bottom. As you can see, we always fill them up with some liquid, because this is the liquid that will be then in the channel. So for example, if you would like to inject some material into the cell, then this is where we would fill it into the cantilever. And then the device is just picking this up And from that point on, everything happens through the software. We use these normal bioplates that anybody has in the lab. So it is quite well adapted to biological environments, let's say. So I talked about injection already, when we deliver something directly into the cells. Now this can be either into the nucleus or the cytoplasm. And you can see how this is done. Well, of course, it's an artist's imagination, but this is the basic principle that we enter the cell, and with positive pressure, we deliver something. As you can see, we normally mix any liquid with a fluorescent marker just to make sure that the injection actually happens, since otherwise the viability of this whole manipulation is more than 95%. So if there is no fluorescence, then we don't necessarily see a lot happening to the cell. This is how it looks like in the microscope as the whole manipulation is being done. We can follow it in real time, and you can see the cantilever kind of squishing these cells. And during, after the squishing, a positive pressure is applied, and then this fluorescent liquid is going to enter the cells. And when we switch into the fluorescence mode, then you will be able to see that indeed the material made it into the cell. Now, one of the big advantages of this method is that it's automated. So the only thing that we have to do really is to select these cells and then aim for the nucleus, the cytoplasm, wherever we really want. And then it just runs on itself. So it's quite comfortable to work with, in my experience. And so this is basically how the injection looks like. Now, what can you inject? Well, it depends on what you're interested in. We have people injecting proteins, antibodies, RNA, DNA, plasmids, even nanoparticles, extracellular vesicles. Anything that comes out of a 600 nanometer hole, more or less, should be fine. Now, from the injection, we can also look at the picking and placing of cells. So in this case, what we really do, as you can see in the little video, is we approach the cell, we grab it, and then we go into a different container and we put it down. Now, in this example experiment, we made an array of 9 cells next to each other, which it looks nice, but there are other applications for this. And one of the main ones is to sort cells monoclonally. So as you can see, we constantly have feedback from the microscope, which means that we always know which cells we are sorting. And especially when people want to edit some genes, normally with CRISPR, Cas9 complexes, then it can be very important to sort the cells that have been edited to make monoclonal cultures from a polyclonal edited culture, for example. And indeed, this is something that we do in a very particular way, because as you can imagine, if you have the monoclonal sorting and you have the injection of CRISPR-Cas9 complexes, then if you put these two workflows together, then what you have is already cell line development. And to do cell line development with FluidFM, this is the road that we follow. And it starts with monoclonal cells. So this is a big difference compared to normal, more conventional methods, where we do editing in batch, for example, with electroporation. And then a certain number of cells will be edited after we have to sort them out. We have to look for some of these edited cells and we have to isolate them. Now it turned around the entire process and we actually start from monoclonal cells, which means that we take a 12-24 well plate and we just put a cell in the middle of each of these wells. And then these are going to be the cell lines at the end. Then what we have to do is to edit them, which is a simple injection. Now this injection, of course, has a certain probability to make the edit, not 100%. But out of these 12 cells, we are going to have definitely a few of them edited. Once the injection has happened, the only thing that we have to make sure is that the cells grow out into actual colonies. And this is more or less waiting around. And of course, there are different ways to speed this up and to make sure that the cells survive growing out from a monoclonal culture. And once the grow out is complete, then we can do the genotyping to make sure that the edit happened where we want, look for some off-target effects, or really just sequence the genes of interest and the loci of interest. And the big advantage, again, of this method, there are multiple, but I would like to emphasize two. So one of them is the general challenge in CRISPR-Cas9 transfection, or gene editing, better said. How do we deliver these molecules into the nucleus? Because of course they have to be in nucleus, they have to come into contact with the DNA in order to take effect. And of course, there are different methods for this. The most popular ones are electroporation and nucleofaction, which do work to a certain degree, but many cells find them quite dangerous. And in our approach, however, what we really do is we go into the nucleus mechanically and directly deposit the molecules into the nucleus. Again, here, we almost don't care about what is the cell type, since from a mechanical point of view, most cells are just bags of protein, and we can pierce these bags of protein and then insert the material into the nucleus. Therefore, this method works with many different cell types, even very sensitive cell types, including some primary cells or stem cells, which would be quite difficult with more brutal methods. So the gentle but direct reaching of the nucleus is one of the main advantages here. And also, as I've already mentioned, we don't need to screen a polyclonal culture, which can be very challenging sometimes. Not always, but sometimes when the edit is such that, for example, fluorescent labels are barely expressed, it is very difficult to screen these pools of cells for certain edits. And we don't make the screening better, we simply omit the screening. There is not a lot of need for that here. Starting from monoclonal culture speeds up the process significantly, which means that we can edit different cell types and also with a much higher speed than what would happen normally. Okay, so this is what can happen if we go into a cell and apply positive pressure. That's really what it is. We inject into a cell through these little pyramids. Now, let's see what happens if we go the other way and we go into a cell and apply a negative pressure. So we take some material out of the cell. This is what we would call a single cell biopsy. The way it works is that we go into the cell once again with the pyramid. We literally touch inside of it. And once we are in the cell, either in the cytoplasm or the nucleus, well, of course, the cytoplasm is more gentle to extract material from. Then we will apply negative pressure. And then the cytoplasm then simply enters the channel inside of the cantilever, and it stays in there. Now, before we go into the cell, then we would normally pre-fill the cantilever from the front with some amount of buffer, with 1 or 2 picoliters. And behind that, so what the cantilever is originally filled with is a mineral oil, which makes sure that when the cell extract enters the cantilever, then it doesn't get mixed with anything except for the buffer that we loaded the cantilever with. So in this way, we can make sure that this extract is not going to, so the valuable things that we're looking for in the extract are not going to diffuse away in the channel, since this mineral oil is really closing the volume that this solution can take up. So once the extraction happened and we have the extracted material from the cell mixed together with the buffer, then we take the cantilever out, and then we can dispense this extract in different droplets, depending on what is the application, really. But basically, this is the basic principle. And what is very interesting is that the cells generally don't mind. So it is quite easy to extract a few picoliters, up to 2 picoliters, without compromising cell viability at all. And we can also extract more. And the cell viability still is very high. This stands for the cytoplasm. Of course, if we go into the nucleus and just suck out half of the genome, then the cell doesn't stand a lot of chance. Now, why is this good? What can we use this for? Yes, we already saw how the pyramidal tip looks like, so that's all right. And actually, this is how it looks like in the device. So we constantly have a visual feedback of how much liquid we are extracting. So this is something that helps us make sure that the cells survive, because we constantly look at how much of the cytoplasm has been extracted. As you can see, it's really in the order of a few picoliters. So a big advancement that this method was used for is what is called live sequencing. You can see the bioarchive paper there in the bottom. So this study has been done here at ETH, so in Zurich. And what the authors did is to extract some material from cells with the FluidFM 30 minutes before exposure to LPS and 4 to 5 hours after exposure. And they could characterize the transcriptome of the same cell through a modified SMART-Sec2 sequencing protocol. Now, the very revolutionary thing about this study, of course, is that it's not just single cell, it's sub-single cell in a way, since we could characterize the transcriptome of the exact same cell multiple times. So even a normal single cell sequencing method, which has the sensitivity to sequence the transcriptome of single cell, would at the end kill, of course, lyse the cell. But in this way, we have the possibility to not lyse the cell, instead really just take out a certain amount from the cytoplasm that then the cell can regenerate and go on as if nothing happened. And then we can really take transcriptomic measurements in different points in the cell's lifetime, which makes it possible to really follow the trajectory in the transcriptomic space for one single cell. And how does the FluidFM Omnium fit into this picture? So this device that I have showed you before with the video. So what that part can do is the FluidFM sampling. So sampling of the cells together with the live microscopy, so measuring the volume and characterizing, looking at the cells if they are okay. And then this little extract that was taken from the cell can be deposited into droplets. And then from that droplet, it is basically up to anybody's creativity to find out what we can get, what is the information that we can get from the droplet. In this study that I have showed you, it was a transcriptomics. But of course, there are also proteins in that little extract. There might be organelles. There might be many different things that can be characterized. So this is for us a very interesting direction in the future. What can we use? What anybody can use these extracts for, in terms of really observing how the cell's trajectory goes over its lifetime. Depending, do we do some kind of treatment or does it differentiate? What kind of events happen during its lifetime? We can really characterize the same single cell when it comes to its cytoplasmic contents. Of course, the challenge is enormous because the content, the biopsy is tiny. It's a few picoliters. And then it's really taking cutting edge methods to characterize these few picoliters of extract. So to sum up, we have fluidic force microscopy, FluidFM, which is a combination of microfluidics and the force-controlled cantilevers. The technology is inherited from AFM, and we can do very different applications, from CRISPR cell line development all the way to omics measurements on cellular extracts. And I still didn't talk about applications in mechanobiology, virology. Of course, we can also manipulate viruses with such cantilevers, and a lot more. And really, it's up to creative people with a discovery mindset to bring out the most of this technology. So if you are interested, of course, welcome to visit our website, where you will find many different ways to contact us. And we are always happy to just chat, receive any feedback, any ideas, any doubts about the technology. So reach out, and you always find someone to talk to. Thank you.